All right. I am John Beckmann, Professor John Beckmann, Dr. John Beckmann, the father of Cid, the transgenic mother of Cin, your hostess with the mostess. If something smells siffy, stop taking a whiffy. Get a green screen in the back. I drink my coffee. Oh, shit, black. Have my brain tied behind my back. Just to make it fair. That's what happens when you have half your brain tied behind your back, just to make it fair. That's proof that I do. Oh my God. Today's a special day. This week is a special week. There's a really cool paper that came out that I knew about it in a sense that I knew the research was happening, but I had no idea when it was coming out. And it kind of hit me by surprise, which is fast. It's interesting. I'm super excited about it. So here's the paper. And this is a collaborative work with senior authors Mark Hochstrasser and Haitao Yang. And Haitao was a postdoc at Yale in molecular biophysics and biochemistry, I think, when I was there. And obviously then I was in Mark's lab when I was in Yale. So we love Mark's lab here. We love Mark's lab on the Beckmann channel. And then of course, my friends Hong-Li Chen, one of the greatest people on earth, and where's Burke? Jason Burke, authors on this. So just shout outs. Super excited to see this work come out. They're characterizing the structures of the proteins that I discovered. It's I don't know, it's like it's an emotional day. It's a cool day when you can actually visually see the structures of the protein that's become so important to you. It's a cool thing. I think I was surprised to see this come out in PNAS. I think that the impact of this is probably higher than PNAS. I think they got undershot. Well, not undershot. I'm sure they shot high. I think there might have been some robbing here. Maybe not. Edit that out. What I should say politically is this is a good paper for PNAS. PNAS got a good publication here. I think that this is going to be a highly cited paper moving forward. I'm super excited to read it. So here, this is my first reaction. I have not read this yet. So let's read it together and let's get my reactions. So the title, Structural and Mechanistic Insights into the Complex, is formed by Wolbachia cytoplasmic incompatibility factors. So obviously, if you didn't know, CI is a phenotype that induces sterility in insects. There's a rescue effect where it can be rescued by females that have a Wolbachia infection. So it acts as like a toxin antidote system. It's a good analogy. And it's run by these two gene operons, which we call CIDs, CINs, or CI factors. Some use that name. Some call it by another name. We like CIDs and CINs because it discusses the biochemical features of these toxin genes. The B gene is always the second gene. The A gene is always the first gene. And usually the B gene has some subset of domains, either nucleases or dubs or both combinations, things like that. That's what you find in the second gene. So we knew that these, it's been known for a while now, since the 2017 Nature Microbiology paper, that A will like bind B. Like there's this interaction. We've known that happens. That was originally established by pull down experiments. But the interface of the interaction was completely unknown. So in biochemistry, you can identify through crystal structures or structural procedures the actual amino acid residues that contact each other and form binding interfaces. And this is important for mutagenesis studies, and it's also important for understanding the functionality. And in this system, it's very interesting and applicable also too, because in this system, what happens frequently in Wolbachia is these 2 gene operons get duplicated and they form these paralogs, which then co-evolve and diversify. And you get A's co-evolving with B's, forming these kind of like lock and key pairs. And as they diversify, they can form these bidirectional incompatibilities where you have one Wolbachia that can't rescue the CI of another Wolbachia and vice versa. And it's always been a quandary or a question about what is the mediation of these bidirectional incompatibilities? How does that work? And the lock and key mechanism has been the best hypothesis explaining how you can get these bidirectional incompatibilities in the context of rescue of Wolbachia. And that now we know is going to be directly mediated by evolution at these interfacing sites. So knowing the sites of the interface will allow bioinformatic evolutionary ecology-based researchers to look at those specific residues in the context of coevolution of A's and B's and in the context of bidirectional incompatibilities to try to figure out actually the genetic justification, the genetic architecture that's underlying bidirectional incompatibilities. So that's one kind of like pitch about like why this is so important for cytoplasmic incompatibility research. So I'm pulling out things I want to talk about. All right, let's digest the intro. Essentially, like aside from all the boilerplate Wolbachia stuff, there is a good discussion about the mechanistic models of CI. And these are essentially like variants of like timing models. These are also, I guess, more closely related to kind of like mod rescue models. Although like mod rescue is so broad that like modification could be the deposit of a toxin in some sense. So that's kind of very broad. But in general, like the idea of the mod rescue hypothesis was that if you had sperm, okay, and in that sperm are chromosomes, right? The mod rescue idea was the idea that Wolbachia makes some factor X, and X interacts with the chromosomes in some way that causes some kind of a change, maybe a structural change, an epigenetic change, a replacement or a loss of a particular protein or something like that. Essentially, like it changes something on the chromosome. And then the important feature here is that essentially like X could disappear and that modification would still remain on the chromosome. So essentially like Wolbachia would like secrete something, it would somehow like modify epigenetics of chromosomes or change or by loss or add something or just subtract something from the chromosomes. And then what would happen in essentially like that modification would then cause sterility. Now, what would happen in the context of an egg? Like once that sperm fertilized an egg, the idea was that the Wolbachia in the egg could produce some factor Y, okay? And this factor Y would then be the rescue factor. And the rescue factor would essentially like be the inverse functionality of the modification. It would essentially like fix the issue and repair the chromosomes. That is the idea of that's like the big summary of mod rescue like ideas. Okay. Now there's various like mechanistic thinking of how a mod rescue model plays itself out. One of these, which is very popular, is this mistiming model. The mistiming model is essentially like suggesting that the modification that happens to the chromosomes on the sperm is not necessarily something that permanently like destroys the chromosomes. It's something that just like alters or slows down the timing of condensation. And so then in the zygote, the cell cycles of the paternal chromosome, so from the dad versus the mom's chromosomes, is essentially just like out of sync. The idea is that like these ones are maybe slower. They're slower to condense. And then essentially like that's what causes the problem. And so the rescue factor in some of these models could have kind of different functionalities. So I just suggested that one case would be the rescue function of the rescue factor, say Y, rescue factor Y, is that it could be in theory like the inverse functionality of the modification and just fix it. And that would imply that it like speeds up under a timing model, it would like speed back up the condensation of the dad's chromosomes to a proper speed such that they become now back in sync with the female's chromosomes. There are other ideas. For example, there is another scenario where the Y chrome or the Y rescue factor could also induce kind of like a similar modification on the maternal chromosomes and essentially like slow them down to like essentially get the timing back correctly. So the mod rescue model has sort of like, again, there's a lot of like vagueness. There's a lot of vagueness and flexibility, and we don't really know precisely which of these is correct. Now there's some evidence, like I'm going off my head here. There's some evidence that suggests that there is some evidence that suggests that the mistiming model could have some merit. And if I remember correctly, I don't remember the exact citation, but there are some studies, I think, where if they essentially like watch the eggs for a longer period of time, I think they do observe that there are situations where the dad's chromosomes can kind of like catch up in situations where you can kind of like, I guess, prevent the mitotic division. I think there are papers that suggest that dad's chromosomes can like catch up, which would be a suggestion or evidence supporting this kind of like mistiming idea. Okay, that's one of the models. Okay, and again, so this gets very nuanced, very detailed. So this might be interesting for some, not so interesting for others. I think this stuff is pretty interesting. The titration rescue like variant of the mod rescue is the idea that whatever the mod factor is, mod factor X, is essentially like adding the thing in. It's adding the modification in, and then the rescue factor Y could again do two different things. It could subtract that titration thing out of the paternal chromosomes, or it could also like add things back into, it could add things back into the maternal chromosomes in a way that, again, like kept the timing accurate. One reason why I don't like the titration rescue model, I don't like this model, because there would be no need to have a two-gene architecture. There's no need to have two genes in the CI operons if the titration rescue model or the titration restitution model is correct, because all you need is one factor X in the male to do the timing modification, and that same factor X can do the timing modification in the female. So if the timing restitution model was correct, I think the architecture of CI would just be one gene, X. But that's not what we see. The architecture of CI are two gene operons, which suggests that there's two functionalities, two separate functions to the induction of CI and the rescue of CI. Okay, so I do not like the titration restitution model of this modification rescue system, and I don't even really like the modification rescue system if you're going to back me into like a corner. I think the timing thing could have some merit, but I still don't like this hypothesis, and I'll explain what I think is a better hypothesis. The goalkeeper, I read that paper like a while back. I just remember thinking like the goalkeeper was like a bizarre hypothesis. I couldn't even like understand it. I would have to like read that paper a few more times to really like understand what the hell they were saying. But I thought when the first time I read it, I was like, this makes no fucking sense. I couldn't understand it. And I'm somebody who's like deeply invested in CI. So if I can't understand it, like I can't imagine what other people were thinking. Okay, now let me explain the other mechanistic hypothesis, which is the TA hypothesis. Okay, the TA hypothesis is a little bit more mechanistically specific. Okay, so the TA hypothesis inherently implies that there is an antidote and that there is a toxin. Okay, and the way that CI would be induced in the TA system is the toxin itself. So in this case, Cin B or Cid B would be deposited into the sperm, binding to the chromosomes or whatever. So if we draw those chromosomes back, we have the chromosomes of the male. These toxins would be deposited essentially like in a region near where they're ready to like fire their toxic activity. Okay. Again, this is different from the modification because you're not modifying the chromosomes yet. You are depositing a factor, a Cid B or a Cin B or a Cindy B, that essentially can like explode the cell when it activates. So it's a little bit different. But like I said, you could interpret this as a modification rescue because the modification could literally be the deposition of Cid B, etc. Okay. But that's how it's a little bit different. It's not like adding something epigenetically yet to the chromosome and titrating it away for rescue. Is a little bit different. This could still fit with the timing model because the function of Cid B or Cin B could in fact be to slow down the cell cycle of the paternal chromosomes. So this is not incompatible with the mistiming thing at all, which is another reason why the mistiming thing does not bother me. I just don't think we have enough clarity yet to know what's going on. Aside from the fact that there are these toxins and the nuclease and the dub functionalities are key to the induction of that process. Now, in the rescue of the TA model, this is really where there is a pretty big difference between modification rescue hypotheses like the mistiming or the titration restitution versus the TA model. In the TA model, there is a corresponding antidote that's produced in the egg, Cid A or Cin A, okay? And there's this lock and key binding where Cid A can recognize Cid B. And so it's essentially like repackaging up the toxin, preventing it from firing. So what the TA implies is implies a couple things. It implies physical binding between the toxins and the antidotes. And this has been proven many times now in all of me and Hochstrasser and Chen's papers, and now this paper. Like the physical binding is a fact. That's been proven multiple times. It's conserved amongst the Cids and the Cins. So we know that that's a fact. And so again, this is why the titration restitutions and even like the modification idea is really kind of a bad idea at this point, because it seems really like rescue happens from a physical interaction. Okay. Number two is that there's this TA implies that there's this cognate binding. It has to be like that because of the bidirectional incompatibilities that you observe. So if the rescue by binding is correct, you would expect to see a divergent evolution amongst diverse pair logs, but yet a co-evolution internally between A and B where they develop these matching pairs. And this is also proven now in multiple cases from Hochstrasser, Beckmann, and Chen papers, okay, multiple times proving cognate binding. So this is explicitly why modification rescue is a little bit different from TA. TA is implying this physical interaction between a toxin and an antidote for rescue. Okay, so where does the 2x1 fit in this? The 2x1 is essentially just saying that you need, in theory, an A also in the induction phase, okay? And again, like I'm not opposed to certain features of the 2x1, Like one idea that I am not against at all, and in fact, there is data to support it, is the idea that A could have this like packaging function for B. So for example, if you think actually what has to happen in a Wolbachia infected cell, imagine these are the Wolbachia, the CID-A and the CID-B factors have to get secreted. So they have to at some point get pumped out of the type 4 secretion system of the bacteria. They have to go through the cytoplasm, get into the nucleus to affect the sperm. Okay, so that's a lot of stuff that has to happen. And if you have your toxin prematurely firing, it could be beneficial to evolve some functionality added in with A to essentially like package the B toxin so that it doesn't prematurely fire its load. Like that, I think like that doesn't bother me at all. And that to me is how a 2x1 could possibly mechanistically make sense. Although, if we're now going to start to talk about 2x1, there are some major flaws of 2x1. And one of these, I pulled out one of these paragraphs, which is this right here: transgenic co-expression of cognate gene pairs, CID-A and CID-B, or Cin-A and Cin-B in Drosophila melanogaster male germlines induces CI like male sterility. Okay, so let's unpack this. As I reread it, it's not patently untrue. Okay, so that was a little bit too harsh. It's not patently untrue. They're not telling you certain things. Okay, it's only true. It is only true for W mel. Okay, it's only true for the W, the CID-A, CID-B, W mel. Okay, if you express, now here's where we get into some huge nuance. If you express Cin-B from W-PIP by itself, you will see weak CI. You'll see weak CI. Now, in this case, it will strengthen if you add Cin-A, but you can get CI by itself by transgenic expression of Cin-B W-PIP, okay? Furthermore, there are other unpublished things. You express them, B factors, okay, so XB, YB. You can produce CI by these Bs alone. And I'm talking like complete CI. This is some of my unpublished data. I'm not going to talk about other people's unpublished data, but I can talk about my unpublished data. This is the idea that you must have A plus B to induce CI is complete bullshit. Okay. And just so that people know, in these factors where you're expressing B alone, you can also rescue it by transgenic expression in the mother of the A. Okay. So it's total bullshit, the idea that you must have A plus B to induce CI. Furthermore, there's yeast data where all you need is the Bs to induce the toxic functionality of the CI models in yeast. So again, the idea that you need both A plus B in all cases is total bullshit. The only place you need it is in one case, W mel, okay? And in the next 6 months, the 2x1 is completely going to collapse because there's going to be a lot of publications where B alone is inducing CI and A is rescuing it, which is proof of the TA model. And I should say that the first one of these that's going to come out, and I'm not revealing anything that's not public knowledge. I'm not involved with this at all. There's a Harvard study from Flaminia's lab where they express, and I know this because it's a preprint, it's a published preprint. Okay, they express CID-B from W-PIP, which is the one I discovered. They express that one in mosquitoes, and it alone is sufficient for induction of CI, which is what we've been fucking saying all along. Okay, so the 2x1 is collapsing in the next 6 months. I wouldn't put all your marbles in that investment. Anyway, that's how these models fit in. But let me come back here. I got a little bit angry there. Let me come back here and offer some consolation. I do think there are some parts of the 2x1 that can have merit. One of which is this idea where A might be important for packaging B in certain contexts. And we have to keep in mind that Wolbachia are not all the same. And Wolbachia that live in different insects are not all the same. And insects have such diverse sexual reproductive systems and such diverse sexual, essentially like sexual what do you call it? Sex determination systems. And the idea that all the CI factors are going to behave exactly the same in all these different insects is patently absurd. You're going to see some differences and you're going to see some divergent evolutions where you're going to see some unique functionalities popping up here or there. But it's going to be a mistake to apply those unique cases as like a model that is supposed to fit all the cases of the insects. And it looks right now, if you actually have an understanding of all the data, that 2x1 is only true for WMel, and it's not true for about 3 to 4 to 5 to 6 other CI factor systems. Okay? And you'll see that come out in the next 6 months. You can bank on that. So the way that this paper plays out is they get the crystal structure. From the crystal structure, they can derive the interface of A to B, and you would essentially generate a list of residues that are in very close contact. And then the way that this plays out is they could then mutate those interfaces. And through pull downs, you can test whether the interaction is eliminated and thereby prove that interface residue is in fact an interface residue. Beautiful pictures. All right, so here are the structures of Cin A, Cin B, and the complex. Here are the interfaces, and these are the amino acids that are important for regulating those interactions in the Cin B and the Cin A. Here's the interface 2, and here's the interface 3. So if anybody wants to study coevolution of A's and B's using bioinformatics, or diversification of paralogs using bioinformatics, or trying to predict bidirectional incompatibilities, these are the keys, at least in the Cin system. And it looks like all the strong interactions are plus, minus, or minus plus charge interactions, which is interesting. So what's worth pointing out in the structure are in Cin B, when you draw it out as like the architecture, the gene architecture, you see the N-terminal nuclease domain, or I like to call this nuc1, and the C-terminal nuclease domain, and I like to call this nuc2. I think it's just a little bit easier. Oh, and they did that too, and D2, and D1. I see they did that too. So they're kind of switching to the nuc and idea. I like that better. NTND, CTND is just a little bit much to say. Here are those two nuclease folds. Okay, here they are. They're a little bit angled. It looks like they're a little bit angled a little bit differently. Here they are. And one of the things that they start to discuss, which I think is going to become more important here in the future, that's one of my predictions, is that there's a little bit of, they're not the same. There's a little bit of diversification in the nuc1 versus the nuc2. And if you look at Tangled Web, which is Joe Gillespie's paper, he actually maps out the consensus sequence, the consensus sequence of many of these separate nuc1 versus nuc2 domains, and they're different. Like so one hypothesis is that if they're different in the amino acid structure, they're doing different things. So the idea, it might be subtle different things, it might be subtle recognition things, but the idea that they're both kind of like I think I originally thought, oh, they're both the same thing. they're both the same thing, and maybe they're like dual functionality, robustness. They cut the same shit. I think that's going to change coming forward as we learn more about the system. I think one of the key things to research now is what is it about these amino acids in these structures that's conveying different functional features of the nuke 1 versus the nuke 2, and how does that play itself out in CI, but not only CI, other reproductive parasitism mechanisms. I think that's going to be interesting. I would have liked to have seen this thing popped on this thing with the electron density clouds filled in. I would like to have seen that. That would have been a cool addition. Here's the schematic cartoon. So, and here's the schematic cartoon suggesting precisely what I was pointing out with the residues. There's these charge interactions. And then it looks like on interface 3, some hydrogen bonding, but I don't know why in interface 3 here, there's charged stuff. What's the deal with that? Let's try to dig into this. So we have a GST, Cin B, and they're making Cin A mutants. So if this band disappears, the binding is ablated. I see. I finally fucking understand. This is like obliterated, where they mutated inverted every charged residue at every interface. Okay, so this is like the negative control, like obliterate binding. And here's what obliterated binding looks like, and the pull down. Okay, this little remnant band that you see, this is GROW L. It's a band that's binding to the non-specific binding to the GST. So this is obliterated control. Then once they have the obliterated negative control, then they start trying to mutate individual interfaces. Okay, so each of these interfaces is where they have inverted all the residues in that interface. And what they discover is that individual interfaces alone, 1, 2A, 2B, or 3, are not sufficient to obliterate the binding. So then they start to mutate multiple interfaces, okay? And then you start to see some reduced binding. So 1 and 2A, a little bit of reduced binding, and then here is pretty much obliterated, and here is pretty much obliterated. So what they've discovered here is that this interface is pretty tight, and if you just mutate one thing, it's still going to stick together. You have to essentially mutate more than one interface to get them to rip apart. And I think the Cin A, Cin B ITC data from the Chen paper did suggest that it was a very tight binding. So this makes sense, that the interface would be rather quite large. Now, this is interesting for the evolution of bidirectional incompatibilities, because one of the things about bidirectional incompatibility is you don't have always, I mean, a lot of times you do have black and white bidirectional incompatibility, but there are bidirectional incompatibilities where you get this like partial rescue. And that could be an effect of, right, like an interface that is weakened, like this 1-2A mutation. It's a weakened interface, which could give you a weakened rescue. And they could, in theory, like correlate in a one-to-one kind of like analysis. It also suggests that developing a bidirectional incompatibility is not going to be just one mutation. It's going to be a series of an accumulation of divergent mutations that progressively lead to a complete bidirectional incompatibility. So you're not just going to get a mutation and then be incompatible. You're going to slowly diverge into partial compatibilities and then go to complete incompatibilities over a longer period of time and mutagenesis. Okay, and then this K is just the reciprocal, where here they have Cin A and they're making Cin B mutations. And I'm assuming that this one is then the obliterated B, which you see no band here. So that makes sense. And this is the one mutation interface 1 and 3 that's pretty much sufficient to ablate most of the interaction. Good data. So they have it with the pull downs, the interface, and they have it with the crystal structure, the interface. So there's 2 convergent data sets, and now they go into the third convergent data set, which is the yeast tests. So here's the yeast tests. Here's the toxicity control. Here's the rescue control, repeating what we observed in the past. And then here's the ablation, the obliterated A, which obviously doesn't rescue. And then here are some of these more minimal mutations. This is nice. So if this is correct, this is nice. They have an interesting partial effect here. So they have a partial rescue at 1, 2B. What you would expect to see there is partial binding. So let's check that. 1, 2B. That's one of the, you can still see a little bit here. And this, it's debatable, but this looks a little bit less. So 1, 2B looks a little bit greater than, the band there looks a little bit greater than 1-3. That's debatable, but there is definitely like a little bit left there binding on. And in my eyes, this one is greater than this one. And so you would expect this rescue to be a little bit greater than this rescue. Let's check that, which is in fact the case. So that's really nice. So they have the 3 data sets there, and then they're doing the same thing here from the reciprocal angle. That's so nice. They have 1, 2, and 3 convergent data sets that all are completely different methodologies that are evidencing this interface. So just really strong data, really strong, elegant design, really strong convergence here from these data. Very nice.
I shall meet death head on. Negate, you've got to get out of here. Beware foul demons and beasts. There are many foes here. The time has come to cleanse my home.
All right, I need to take a break.
This monastery reeks with evil and corruption. Oh, I hate staining my hands with the blood of foul sorcerers. Eternal suffering would be too great for you, Diablo. Let the gate be open to die. I shall meet death head on. I shall meet